Right, hi guys, as promised, this is the video that accompanies going over your tracking point 1 or your mock paper. As we go through, I'm going to hopefully explain the answers in a bit more detail. Feel free to skip questions that you felt confident on or that you feel like you don't need support on, but I think for lots of you, even just going through the theory behind lots of these questions will help. Pause at any time. At any time you want to ask a question, if we're in lessons while you're doing this, stop, stick your hand up and I will come over and explain in a bit more detail for you. So we're going to get started straight away with question 1. So the beginning of this question is hopefully something that we should be confident with, and this relates explicitly to topic 1. And we should hopefully be aware that the primary structure is before the protein begins to fold. So my primary structure is made of, and these are the mark points, a sequence or order of amino acids. That's the first mark. And we have to specify the monomer. Okay, that single repeating unit that we see over and over again. And we have to specify as well that they are joined by peptide bonds. There is no further detail that you need in this question apart from the fact that it is the order or sequence of amino acids and they have to be joined by peptide bonds. We are in A-level now, we are not in GCSE, and therefore you cannot just say they're joined by bonds, or joined together. You have to be specific and concise in what you're trying to explain. Now question B is about the genetic code, and here we're moving into topic 4 territory. These 3 keywords we see over and over and over again in the specification, in the textbook, in any online resources that you use, so you have to be confident in understanding their meaning. Now the first one, universal, is a little bit of a misnomer in my opinion. Universal to me means that it's everywhere, which yes, that's true, but saying that the genetic code is everywhere isn't going to cut it for this answer. So here, in terms of the universality of the genetic code, you needed to make sure that you've said the same codon, okay, or you could have said triplet, codes for the same amino acid and then lots of you, which was really, really great to see in your answers, put something like regardless of the species or regardless of the organisms. So let's say we've got the triplet codon CCC. If, let's say that this produces the amino acid methionine, that same triplet codon CCC is going to produce methionine in E. coli bacteria, in whales, in a holly bush, in a bird, like a hawk or something. That's the whole idea, that all organisms, regardless of their species, regardless of their type, are going to have the same amino acids coded for by that single triplet alone. Okay? It allows us, when we look into topic 8, to start looking at recombinant technologies and transgenic organisms. And so that's really, really key that everyone follows the same basic instructions to make proteins. Non-overlapping, I was a little bit disappointed in. I think for lots of you, it was evident that you kind of understood what it meant but didn't know how to explain it. And so let's consider a sequence of bases like this. Okay, so AT, A, CCG, A, T, G. Now the whole idea of non-overlapping is that each one of these bases is only read once. Okay, so there's no overlap. And in the mark scheme, the way you needed to explain this is that each base is only part of one codon. Okay, so you can see here the A, the T and the A are read once. Immediately then I move on to the C and the G. I don't consider ATA as one and then go TAC as a second. Okay? They are non-overlapping, discrete units of 3 bases that once we've read those 3, we don't read them again. Okay, so each base is only part of one codon, or you could have said that codons do not overlap. And I know this second answer, this second mark point, seems a little bit silly considering, you know, the word that you're defining is non-overlapping, but sometimes the answers are as simple as that. And finally, degenerate. This was a word that was defined quite well across the class, and that it's more than one codon codes for the same amino acid. So if I continue with this example, I know it's not correct, this is just an example. Let's say CCC equals methionine. We could also see that ACG makes methionine as well. And the idea of the degeneracy is that methionine will be made by multiple different codes. Okay, there are only 20 amino acids, but I believe there's 64 different permutations of these bases. Right? So we've got to somehow make 64 different triplet codons into the 20 amino acids, and that means that there's going to have to be more than one triplet that goes with each of those amino acids. Okay, I'm done with question 1 now. So if you need a little recap, you want some time, here's the questions with the answers zoomed out. Pause the video now, and then we can get on again in a second. Okay, question 2 then. And here we start seeing evidence of you guys not reading the stem properly. So in this question, we're discussing the human papillomavirus, which contains double-stranded DNA genomes. Okay, this tells me that it's already got a human-like genetic material, or an animal-like genetic material even. And which components are found in a human papillomavirus? Well, we know that because of our viral structure, which typically looks something like this. So we've got this capsid, we're going to have some attachment protein legs, there's going to be some genetic material on the inside, there could also be some enzymes, for example. We're always going to have capsids. We're always going to have attachment proteins. So these are all fine. However, the difference comes if we're considering the role of things like the cell surface membrane. We know that we don't talk about viruses like we do other prokaryotic cells. Viruses are not alive, they're not considered prokaryotes. And so they don't have the cell surface membrane. They don't have that plasma membrane as other cells, or cells, not other cells, would. Equally, the capsule, we generally talk about a slime capsule with regards to things like bacteria. Okay? This is not a bacteria. We've been told it's a virus, so we're going to get rid of that second one. And lots of you were fantastic at identifying that the answer was either one of these two. Now, there's a very specific reason why we do not need reverse transcriptase. And lots of you in your answers wrote, yes, correct, that RNA is converted to DNA, or capital D would be helpful, DNA by reverse transcriptase, and that is true. But that's for HIV. Okay, this is where we see the genetic material being changed from RNA to DNA in HIV and in retroviruses. However, in this example, our genetic material is already double-stranded DNA. I don't need that reverse transcriptase. I don't need to turn my RNA to DNA because I don't have RNA. I've got DNA already. And for that reason, the top answer, capsid and attachment proteins, is the answer that you should have given. Now, the DNA replication enzymes of a human cell makes copies of the human papillomavirus genome. So let's just double check what we're talking about here. DNA replication enzymes. Okay, let me think about it. This is topic 1, and I know that with DNA replication, I might be considering helicase, DNA helicase. I might also be talking about DNA polymerase. And so these enzymes of a human cell make copies of the human papillomavirus genome. We know from the stem on the previous page that this is already DNA. So we're going to have to name 2 enzymes that are involved in replicating the DNA. Oh, we've already done that. Lovely. And describe their roles in the replication process. So let's have a look at this first mark already. I'm going to put number 1 here because these 2 go together. You had to mention both of them to get these marks. We're talking about DNA helicase and DNA polymerase. The second mark is for giving the role of the enzyme DNA helicase. And so for that role, you needed to say that helicase is there to break hydrogen bonds. Okay. You could have said break hydrogen bonds between complementary base pairs. I'm just going to put comp BP here. Equally, you could have said it unwinds and unzips DNA by breaking hydrogen bonds, but this breaking hydrogen bonds is the mark that you needed, or the point that you needed to get that mark. For the third mark, you needed to mention the function of DNA polymerase, and lots of you made a bit of a boo-boo with this one. Lots of you decided to tell me that it's involved in something like complementary base pairing. That is not the case. So what we see happening in DNA replication is we've got our template strand. I'm going to put here template strand of DNA. Okay, and we've got our exposed bases, A, T, let's just put them all down, C, G, A. And what we get in DNA replication is we get free nucleotides in the nucleoplasm just floating around that come over and find their complementary base pairs. Okay, so these are all individual nucleotides that have got their phosphate, I'm going to put it here, and their ribose sugar. Okay, you get the gist. So they've all got their little pentose sugars, or hexose sugars, sorry, and the phosphate that attaches them. And what DNA polymerase, does is it goes through and it joins the sugar phosphate backbone. And so for this mark, you need to say that DNA polymerase helps either, and I'm going to put both marks here, joining nucleotides via a condensation reaction, or you could have said that it forms phosphodiester bonds. And you don't have to specify that it's between the sugar and the phosphate, but you do have to specify that it's between adjacent, so next to each other, nucleotides. So continuing down to question C, we can see now that we're continuing to talk about this human papillomavirus, and it's infecting cells that are no longer dividing. So this could tell us that they have, let me change the pen size, I can't. could tell us that they are already specialised, so they've left the cell cycle, or perhaps they're just about to undergo apoptosis, so that controlled cell death. The human papillomavirus genome contains genes that code for proteins that cause human cells to restart their cell cycles. So we've got the code for proteins. Obviously, we know that these are genes in general that cause human cells to restart their cell cycles. The human papillomavirus infection can cause cancer, and we need to figure out why. So using our knowledge of cells that have left the cell cycle and what cancer is. Cancer is caused by uncontrolled cell division. Now, if this cancer, this human papillomavirus, is causing these cells to restart their cell cycles, that is uncontrolled. You haven't done anything to it. So actually, in this case, that is all you need for the answer. You could have said uncontrolled cell cycle, uncontrolled cell division, I'll put here cycle, division, or uncontrolled cell mitosis. The important thing here is that it is uncontrolled. Okay, that is the key part of this mark. Remember that it's talking about body cells, so human cells. It hasn't specified gametes. Does it say anything in this stem? Nope. Okay, so we're not necessarily looking at gametes here, so meiosis is not allowed. And in the same vein, it does say in the mark scheme to ignore growth. Okay, growth is not the same as division in terms of our cells, so we can't talk about either of those two processes to the right there. Now, the human papillomavirus is transmitted through sexual contact. Okay, so this could be due to fluid exchange, it could be due to contamination, sort of physical contact, maybe of something like warts. And more than 95% of cervical cancers, which only affect females, are due to HPV infection. HPV infection of other tissues increases the risk of cancer. Okay, so potentially here they've specified that cervical cancers are only in females, but haven't specified that these cancers can't be in males as well. So males, question mark? But this is rare compared with cervical infections. A vaccine is available that is over 80% effective, but not 100%, at preventing HPV infection. If given before the person has been exposed to HPV. So this bit is important because it implies that if it's transmitted through sexual contact, if you are having this vaccine before you begin sexual contact, you are likely to be 80% effective or 80% protected against HPV. There is evidence of herd immunity when more than 50% of the population has been vaccinated. So herd immunity is where we've got vaccines for the many to protect the few. And this question is an evaluate. So evaluate whether 10 to 12-year-old boys should be given HPV vaccines. And the key here is this word evaluate. So we need to discuss the pros and the cons. So we need to have a yes and a no and, or a no but. 10 to 12-year-old boys is important, so we need to consider the effect of age. And the fact that it's boys is important because we have to talk about the gender separations. Okay, so we've said already in this stem that cervical cancers are 95%, or more than 95% of cervical cancers, which are only going to affect females, are due to this HPV. So we're going to take a list now of the for and the against points, and I will come back and tell you how the marks are kind of awarded as we go through. This is a 4-mark question. So things for that you need to give it early. I'm going to put V in a circle for vaccine. Early, before, exposure to HPV. And another mark here, or the same mark, sorry, you could have said before sexual activity. We're assuming there that 10 to 12-year-olds are not having sex. Obviously, this is quite a valid assumption to make. 10 to 12 is probably too young. Another argument for the case of using these vaccines in the 10 to 12-year-old boys is that it reduces the transmission to girls. And that's helpful because, remember, we're looking for herd immunity. So you could have also said rather than transmission, spread would have been fine. In other positives or other arguments for, we need to remember that boys still can be infected with HIV. So protecting them is important. Or not HIV, HPV, sorry. This HPV can increase the risks of other infections. And for the same mark point, you could have said that the vaccination prevents HPV infection. I'm just going to put infection and abbreviate it. As well as this, we need boys to be injected. Let me just try and get my injected and infected properly. Injected with vaccine to enable that herd immunity. So if only girls are vaccinated, they make up about 50% of the population. We need more than 50% of these boys to be vaccinated to ensure that herd immunity. As well as this, HPV can cause an increased risk of other cancers in these boys. And obviously, we don't want to increase the risk of anyone having cancer. Famously, cancer is a bad thing to have. Our arguments against are less numerous. Okay, so in these mark points, you needed to get a maximum of 3 of these to get 3 out of 4. Okay, so 3/4 of these 4 marks are awarded for these 4 points. It's very easy to make arguments for in this question, this context. However, against, and lots of you were great at this, the boys are at less risk of cancer from HPV than females, but lots of you wrote the same mark point here. So cervical cancer only affects females. And also, we don't really know what the long-term effects of these vaccines are. It hasn't mentioned anything here. You can make an assumption then that if they haven't mentioned it, they don't know, and that we're not sure of side effects. So could it affect fertility? Could it affect growth? Could it affect anything? We don't know. So you've got to make sure that we're considering all of the perspectives, so the safety, the population impacts, as well as looking at the context of this question, so the age, the gender, and the overall benefits as well. Question 3 is looking at the mitotic index and the required practical, where we looked at mitosis in some roots. So a student prepared a plant root to observe cells undergoing mitosis. Already with this stem, I'm thinking about the fact that plant roots undergoing mitosis, I know that potentially I'm looking at the meristem. Okay, so I'm looking at the root tips because I know this is where mitosis is happening. Also, maybe I'm considering my knowledge from topic 6 with growth factors. And I know you didn't know topic 6 at this point, but for future reference, it helps to be mindful of the wider context of these questions. So he put the root in a small bottle of hydrochloric acid, HCl, in a 40 degree, so that's warmer than body temp, water bath. Why did he put the plant root in acid? And the reason that he's put it in acid is because hydrochloric acid is able to react with the water that is found in solutions that it's in, it's found around cells, it's found in the cytoplasm. And what it does is it enables hydrolysis. Okay, so it enables hydrolysis of the phospholipids themselves, and therefore breaks down the cell walls, breaks down the cell itself, and allows stains and things like that to get into our cells. So it enables hydrolysis of the cell wall. So we're going to hydrolyse those bonds between the cellulose, those glycosidic bonds, and this in turn is going to enable the stain to enter the cell. Other marks that you could have had was to separate the cells, so the individual cells themselves. You could have also mentioned that it is allowing the cells to be more easily squashed. Notice here how I'm saying that it's allowing it to be squashed more easily. So that is to say that we can still squash them without the acid. It's just easier once you've got this hydrochloric acid around. And also, lots of you got the mark to say that we're stopping mitosis. Now, you could say to stop mitosis continuing, you could have said to stop cell division, you could have said to stop the cell cycle. But remember that hydrochloric acid is a solution, it's aqueous, and therefore it can produce the water or can add in the ions needed to hydrolyse the glycosidic bonds between the cellulose cell wall. Now, 2 precautions required when working with hydrochloric acid. Precautions are things that you do to keep you safe. So you need to make sure that you know the risks, the hazards, so what could cause you harm and what's the likelihood of causing harm, and then how to prevent that harm from occurring. The wording here is really, really important. So you could have said for the first mark, eye protection or goggles. As this question is just a state, you don't need to explain. So you don't need to tell me why that eye protection is important. I'm going to presume that you know why eye protection is important. Also, along with eye protection, because this acid is a liquid, you might want to use gloves. Gloves alone is a mark. If you do have spillages, you might want to say you can add water, and you have to say water specifically. That's going to have the effect of diluting the acid and make it slightly less corrosive, reduce the hazard associated with it. And then finally, we're going to make sure that we do not pour it down the sink. Again, these acids can be quite corrosive, so we don't want to put them down any material that is potentially going to corrode as well. So now we're looking at some data. The scientists determined the mean mitotic index, and that is the number of cells dividing in a field of view divided by the total number of cells in the field of view times 100. Okay, so these 50 micrometers, I'm just writing down all of the information that I think is important. Intervals away from the root tip in 10 young plant roots. So we're going to assume that this is an average of all of these root tips. So the graph below shows the scientists' results. On the X, we've got our independent variable, the distance from the root tip, and on the Y, we've got mean mitotic index. So that is our dependent variable, things that we're measuring. The null hypothesis for this investigation. So remember, the null hypothesis, we're talking about the significance of difference. And so lots of you were fantastic at this. I think psychology really helped here. The null hypothesis is that there is no significant difference between mean mitotic index at 200 micrometers and 300 micrometers. Remember, we discussed this in class. We said that for this null hypothesis, you need to say no significant difference between the dependent variable at the independent variable option 1 and the independent variable option 2. So here we've got our dependent variable and our two options, or our two conditions for those independent variables. Now, the scientists recorded these measurements from tissue located at 200 micrometers from the root tip. So automatically I'm looking at my table, my graph, sorry, I just want to have a look and see what my value is. So I'm going to take that as the mean mitotic index. So mean mitotic index is 0.35 or 35%. Use this information and the graph in C to calculate the number of cells undergoing mitosis at this location. Assume there are no spaces between the cells and show you're working. And the first thing I hope that you recognised is that the units for the field of view area and the mean area of one cell are different. And so if you return back to your knowledge from topic 2, you will know that from millimeters, or milli, the prefix, 10 to the minus 3, to micrometers, which is times 10 to the minus 6, I need to times my number by 1,000. But the important thing, and where lots of you went wrong initially, is to realise that this unit is millimeters squared, or micrometers squared. So I can't just go from millimeters to micrometers. Instead, I'm looking at going from millimeters squared to micrometers squared, which means that for the context of my area, okay, so let's say this is 0.2 millimeters squared. If I want to increase or change my units, I have to scale my values by 1,000, so change it by the factor of 1,000, or 10 to the 3, in both the x and the y direction. So let's make this a little bit clearer. If I've got this square that's 0.2 millimeters squared, if I square root it, I find that each side is worth, each side is about 0.44 meters. Let me just double check that on my calculator. 0.2 square root, 0.44. Let's go for 0.45 because it's 0.447. So that means that the x direction is 0.45 meters, and the y direction is also 0.45 meters. And when you multiply them together, you get that 0.2 millimeters squared. Okay. Now, if I want to change this to micrometers, what happens is I've got to increase each of these values, the x and the y, by the factor of 1,000 to change both the units. Okay, if I only do it times 1,000, I'm only going to be effectively changing either the x or the y. So now what happens is that on this x-axis, or the x direction, my 0.45 millimeters becomes 45 micrometers. So I've scaled the x direction by a factor of 1,000, and I'm going to do the same to the y. So this again becomes 45 micrometers. In doing this, what happens now is that my total area value doesn't just increase by the factor of 1,000. Because I've done times 1,000 on the x and times 1,000 on the y, instead my unit, or my number value, my numerical value for this area becomes 1,000,000 times bigger, because we've got that 1,000 from the x, 1,000 from the y, 1,000 times 1,000 is 1,000,000, and so now this 0.2 becomes 200,000. So 200,000 micrometers squared, and this is what we're going to be using to calculate the number of cells. So from here, I know that my area, my total area, is 200,000 micrometers squared, and the cells take up 3,495 micrometers squared each. The first thing I'm going to do is I'm going to see how many cells I can fit in this area by dividing my total area, that 200,000, by 3,495. And I'm only going to work with the numbers now because the units are the same, so I'm going to ignore them. So calculator, 200,000 divided by 3,495 is 57.22. Now this is obviously not a full cell, but that's fine. And if you manage to get to this point already, you will have got 1 mark. Okay, so incorrect answer, but a mark of 57 or 57.2 will have gotten you 1 mark. From there, we needed to use the fact that there are 57.22 cells within this field of vision, and my mitotic index is 0.35. So 35% of the cells at 200 micrometers from the root tip are undergoing mitosis. So I'm going to times that factor, that number, 57.22, by 0.35, which is my mitotic index, to give me my final answer of the number of cells that I expect to be undergoing mitosis, which is, hold on, bear with, 20.027. Answers you could have had were between the range of 19.95 to 20.03. This answer obviously fits, and the range itself is there because of sort of things like rounding. So if you turned this 57.22 into 57, you will have got a different value. If you said 57.2, you would have got a different value. It just gives a little bit more scope for the way that you've worked the maths out. What doesn't change, though, is how you read the graph. So it's very, very clear to me, at least, that this cross at 200 micrometers from the root tip is at 0.35. So please make sure you're looking at the correct data on the table as you go through. The end of this question then is looking at the kind of reliability and validity of your investigative processes. So another student is comparing the mitotic index in the root of 2 different species. So now we are looking at the different species as my independent variable. That's the thing I'm changing. It's a completely different investigation because it's another student doing it. And we're comparing the mitotic index, our dependent variable, from these 2 different species. Give 2 considerations, so 2 things that she needs to consider within her method to ensure this comparison is valid. And validity relates to the ability to say that your results are directly measuring what you set out to. So really, we're talking about what can I control? And there were lots of fantastic answers for you guys, really, really good. So very impressed, well done. And I'm just going to list the marks that you could have had for this because it's probably easier that way. So the first one you could have had is that the roots or the plants were the same age. Equally, you could have said that the roots or the plants were grown in the same conditions. So same growing conditions. You could have also said that the roots or the plant samples were taken from the same distance from the root tip. So if you're going to use 200 micrometers, always use 200 micrometers. If you're going to use 10 micrometers, always use 10. You could have also said experimentally that they are in the acid for the same time or for the same temperature. You could have said that the same concentration of acid. You could have said that we're going to have the same pressure on the squash of the slide. And if you're not sure what this means, refer back to the practical notes. So what you should have done is covered your sample of root tip with a cover slip, wrapped it in some paper, and then pushed down firmly on the cover slip on the slide to produce your squash. The idea is that if you put the same pressure on the squash each time, you will sort of spread the cells out equally on both of them, and then you'll get the same field of view, or the same perspective in your field of view for each. Obviously, the best type of investigation is one that is repeatable and reproducible. So repeatable means I can do it over again. Reproducible means that my results are similar each time. So I'm going to want to do several fields of view for each of these squashes. And then obviously I'm going to calculate an average mitotic index with those. And finally, I'm going to do it at the same time of day. I'm not going to go into how time of day is going to affect our roots and things like that. Just need to know that it is a possible answer. Cool. So let's move into question 4. The human disease malaria is caused by an infection with a single-celled, so unicellular, eukaryotic organism. So the good thing to know about malaria is that it's a plasmodium. It's a kind of protist thing that will infect your cells. And figure 1 shows a diagram of Plasmodium vivax, one of the species that can cause malaria. Here we can see in the figure that we've got structures containing enzymes, and I'm going to write here that enzymes are proteins and they're biological catalysts, so generally they're going to speed up reactions. And these enzymes are there to allow P. vivax to enter the human cells. Now, how are they going to enter the human cells? Are they going to dock with glycoproteins? Are they attaching to receptors? How are they doing it? It's just things to think about as we go through. And so for this question, we have to use the diagram. So it says, other than the Golgi apparatus, or the Golgi, whatever you want to call it, name one structure in figure 1 which shows that P. vivax is a eukaryote. And there are a few options that you could have said. So Golgi apparatus, we are not discussing the organelle that I've just put across through. So here you could have said that we have got a nucleus, nice and easy. And I'm going to put a number 1 next to it here. I can tell it's the nucleus because it's got the nuclear envelope with pores leaving it. Number 2, I could have said organelle that's adjacent to the nucleus, which in this case is the rough endoplasmic reticulum. And sometimes the mark schemes are a bit funny. They won't accept things like RER, so it's better to be safe and write the whole thing. Additionally, you could have had this third option up here, which is the mitochondria. The reason, however, that some of you might not have got the mark is because of the fact that you just wrote the term ribosomes. Okay, and it's important here to clarify that all organisms have ribosomes. Prokaryotes, archaea, all of them have got these ribosomes. If you needed, or if you wanted to get the mark for ribosomes in this question, you needed to relate it to the eukaryotic ribosomes. So you needed to say that they were larger or large ribosomes, or you could have said a specific detail about their size in that they are ATS ribosomes. Okay, they are big, much bigger than those prokaryotic organisms' ribosomes because we've got more complex proteins generally in eukaryotes. The final option that you could have had is this last organelle that I've labelled 4 up in the top. And it says that in the question we've got structures containing enzymes that allow P. vivax to enter human cells. And because of that, I'm going to presume that those organelles that have the enzymes in them are lysosomes. If you've written lysosomes, absolutely fine. but you would have also been allowed to say vesicles for the same mark. So you can't have lysosomes and vesicles. Notice as well that it only wanted one, so if you write more than one, you're just wasting your time here. Then we needed you to describe, so tell me what they do, two functions of the Golgi apparatus in a eukaryotic cell. Now here, the eukaryotic cell is kind of a moot point. It's just give two functions of the Golgi apparatus because prokaryotes don't have a Golgi. And you could have said that we needed them to modify, package, or transport proteins. For the same mark point, you could have also said that they are there to make or transport glycoproteins. Okay, so these are both the same mark point. You're not going to get 2 marks if you've written both of these. Just to clarify other terms that you could have used, for modify, you could have used the term process. You could have also said that you are going to add carbohydrates to proteins. That's fine. And those would make up the glycoproteins. You could have also said that you're adding lipids to them to make them lipoproteins. It does say that you could have, make or transport lipoproteins as well. And as well, just for clarity's sake, for this last mark, you could have also said that we're producing chylomicrons. Chylomicrons we've seen in the digestion and absorption of fats in topic 3, I believe. So if you're not sure about that, please just go and double check. And your second mark for this question is to say that we are going to modify, package, or transport lipids. So the crux of this is that you need to understand the role of the Golgi apparatus in protein synthesis. So in that final step, once we've done transcription and translation, we're then going to send those proteins from the ribosome to the Golgi to start becoming modified, packaged, or transported, put them into vesicles, or they're there to modify, package, transport lipids, and in the same vein for that mark 2, you could have said make and transport glycolipids. The same mark points in terms of like changing the word modify for something like process or add carbohydrate, they still stand. The final mark though that you could have got for this question is that the Golgi apparatus is there to form, wrong colour pen, is there to form or release vesicles. So following the processing of either our proteins or our lipids, we're forming or releasing vesicles or lysosomes. Notice and remember it's lysosomes for the vesicle. Lysozymes are the enzymes that are typically found in those vesicles. So PVVAX evolved from a common ancestor in Africa. Common ancestor, we're looking at phylogeny or phylogenetics. As humans migrated around the world, new strains of PVVAX evolved. PVVAX is now extremely rare in Africa, but there are several different strains of PVVAX in other parts of the world. Figure 2 shows a phylogenetic diagram of the evolution of these different strains. What does Figure 2 suggest is the order of human migration out of Africa? So what I'm doing in this question is I'm putting them in order of when they split away from this common ancestor. And I know from my revision session on phylogenetics that the far right hand side of my time scale is the common, is not the common, is the present day. So if I work back towards the left, the further to the left my node is, that splitting or branching of a phylogenetic tree, the longer ago that division was from the main population, but equally would suggest that the further ago they were dividing away from that common ancestor was the first groups to move out of Africa. So in terms of my time, I can see that this node here, where I get branching off of the Indian strains, is going to be first. Okay, this happens first, which means that I can immediately get rid of the top and the bottom ticky boxes. Following that, my next node, so that separation of a common line occurs here. Okay, so at the top of my phylogenetic tree. And this is where I see the branching off of the European strain. So now I know that I'm looking for a line that says India, Europe. So far, this third one has got my answer. Then I'm going to look for the next branch, which is here, just to confirm my answer of that third box, that third tick box is correct. And I can see that this node follows to the East Asian strain. Perfect. I'm not going to bother doing any more because I've confirmed I've got my first two migration points out of Africa. Great. And then the East Asia one just confirmed that this third tick box is correct. If you weren't sure and you did want to go through, the next node separates Central America, and then finally that last node separates the South American strain from that common ancestor. And this is a maths question that is quite wordy, and because lots of us tend to skim over the finer details of the questions, lots of you got this wrong. So I'm going to take you through and we're just going to highlight all of the numbers. So there are an estimated 229 million, and if you don't know what that looks like in numbers, so 229 and then 6 zeros, cases of malaria worldwide per year. 94% of these cases of these cases are found in Africa. And this automatically tells us that 6% of these cases are not. And all I'm doing as I'm going through is using my logic to separate out the details. So 94% of these cases are found in Africa, 6% aren't. But of these 94% of these cases that are found in Africa, they are not caused by PVVAX. However, PVVAX does cause 61% of the cases of human malaria outside Africa. Use this information to calculate the number of cases worldwide caused by PVVAX each year. And automatically, the fact that they've said worldwide means that I'm not considering Africa. Okay, so I know that of these 229 million cases, 6% of those are found outside of Africa. Okay, these are found in the rest of the world. And if I do the 229 million times 6%, or if I'm writing it in my calculator, 1, 2, 3, 4, 5, 6, I'm going to times it by 0.06. But remember, you can also write in your calculator 6 over 100. That's going to give me an estimated number of malaria cases worldwide of 1374000. So 1374000. And that is, 13,740,000. And it tells us in the question that PVVAX does cause 61% of the cases of human malaria outside Africa. So now, of these 13,740 million cases in the world outside of Africa, 61% of those, or 0.61 or 61 over 100, are caused by PVVAX. And if I smash that into a calculator, I end up with 8381400. So 8,381,400. And I'm going to write that straight into my answer box. So that's a 2. 8381400. But because this is a faffy kind of question, they've got a few different numbers that you could have written. So you could have written exactly like that, You could have written it as 8.3814 million. You could have written it as 8.3814 times 10 to the power of 6. You could have written 84 million, or sorry, not 84 million, 8.4 million, or 8,400,000. Is that the right number? Yeah. Or 8.4 times 10 to the power of 6. I'm not entirely sure why they rounded it to the 8.4 million, but, you know, we're not going to complain if the mark scheme gives you more opportunities to get a mark. Now, our final question for this one is, in Africa today, most of the human population are resistant to malaria caused by PVVAX. And now, automatically, I'm thinking straight away that I've seen resistance in topic 4. Okay, and in topic 4, we had a look at selection. And the kind of selection that resistance is a directional selection. So we're tending to keep the extremes, the ones or the individuals that are able to resist malaria. Use your knowledge of natural selection to explain, give reasons for why this resistance is so common in Africa. And we've got to start from the very, very beginning. Okay, so our first step in any kind of natural selection is that genetic mutations caused a change in an allele. And if you wanted to, you could specify that that allele is for PVVAX resistance, but you don't have to. The second mark then is to say that this allele, not the gene, okay, it's a variation of a gene, means that those, or that carriers, are less likely to be infected or to get malaria or to get PVVAX. You could have also said for this same mark point that they are more likely to survive malaria. I'm going to put that down here. And as a result, we see that these individuals are more likely to survive and reproduce. So, carriers of allele. And we're going to do the holy trinity of natural selection. So we're going to survive, reproduce, and the most important part for this third mark is to pass on the allele, not the gene. Remember, it's the allele, it's the variant that we've got. So over time, because remember we're talking about the change in the population, so the whole of Africa, we see that over time the allele frequency increases. And what this results in is more people having the allele, and therefore more people are resistant to this malaria caused by this specific strain of bacteria. As well as saying that the allele frequency increases, you could have said that there are a higher proportion of this resistance allele, or that the allele for resistance is more common. Either of those are absolutely fine. So from our class's results, we can see that selection and all different types of selection are not necessarily our strongest suit, which is absolutely fine. That's why we kind of go over these papers in a little bit more detail. So the first part of this question 5 is explain what is meant. Okay, so this is really asking you to define. And we're looking at this term stabilising selection. It also says describe the circumstances under which it takes place. So I need to define this keyword and then give reasons or give the kind of environment that happens or that causes this to happen. So our first thing that we need to do is our definitions. And the definition of stabilizing selection is a kind of selection where the extremes are selected against or the mean is selected for. So we can have either the selection against the extremes or a selection for the mean or average trait. And an example of this is, or the key example for this is birth weight in babies. Okay, so typically birth weights in babies show this bell-shaped curve. I'm going to put kilograms on the bottom and frequency on the side. And what we see happening over periods of time or circumstances where my environment is unchanged, so nothing has majorly happened, there's no particular natural disaster, so the environment is stable, we see that these extremes, so either end of my bell-shaped curve, are not advantageous. In terms of babies' weights, these low-weight babies are more likely to die from things like infections and stuff like that, whereas our high birth weight babies might just be difficult to birth and could have complications. So over time, what we see happening is that the spread of a trait that undergoes stabilizing selection, the curve becomes very narrow. All right, and this is what you needed to explain in your answer. So firstly, you needed to say that it occurs when we've got a stable environment. And the result of this is that we see a reduced range of values for the variable. In this case, we're talking about birth weight, or in the example that I'm discussing over to the right-hand side, we're talking about birth weight. This isn't a specific example to this question. There is no context for this question. So stable environment, we're reducing the range of values of the variable, and as a result, we see an increasing proportion of the population being adapted to the environment. So these babies with the kind of average birth weight are more likely to survive because they either don't have difficult births, they're not going to get stuck, and the mother is unlikely to have to undergo things like C-sections, but equally they're strong enough and big enough to maybe not get infections as easily. So an increased proportion of the population are adapted to the environment, whatever the environment might be. Again, it's unspecified in this question, so I'm not going to write anything in my answer about a specific example. I'm assuming already that this is going to come a bit later. And here we've got a lot of STEM, and it's important really to read all of this STEM because there is key information here which are going to help you answer the question much more effectively. So some European clover plants can produce cyanide. The plants that can produce cyanide are cyanogenic. Those that can't produce cyanide are called acyanogenic. Cyanide is toxic to the cells of animals and plants. So already I'm thinking, okay, so cyanide is toxic to plants. Therefore, if plants produce cyanide, can they be damaged by it themselves? So I'm going to just put up here just my thought. Affect the plants that secrete it or that are cyanogenic. And I'm just going to leave that there with a question mark. Okay, second paragraph. When the leaves of cyanogenic plants are damaged by slugs, right, okay, so now we're thinking about slugs, or exposed to low temperatures, membranes within the cells are broken. Okay, so again, next point, membranes broken by slugs or cold temperatures. And now I'm trying to link that back to my initial thoughts. So if my membranes of these cyanogenic plants are broken, are they then going to damage the rest of the plant with the cyanide that's contained in those cells? This causes the release of enzymes that control the reactions which produce cyanide. Here we go. I'm starting to link my thinking now. So the release of enzymes that control the reactions which produce cyanide. Okay, so cyanogenic plants, and I'm just taking this from the question, plus slugs or cold temperatures equals release of cyanide. Fab. So now I'm thinking, okay, just getting some information. I've got that background. Cyanogenic plants are going to cause the release of cyanide, and that is, I'm assuming, going to affect the plant. The proportions of cyanogenic and acyanogenic plants in clover populations were determined in different parts of Europe. These are shown in the diagram below, together with the minimum or the mean minimum winter temperatures. Final bit of this stem, which is really, really important. Slugs are not usually active at temperatures below 0 degrees. So I'm just going to annotate my diagram quickly with this line here for 0 degrees. And I'm annotating it because I need to visualise the fact that anything above this blue line that I've written has no slugs. And anything towards the bottom where my temperatures are getting warmer, so 4 and then 10 degrees, I am going to have slugs. As we can see down the bottom, there's a key. So the black side of any circles, the black portion of any circles shows the population of cyanogenic plants, and the white portion shows acyanogenic plants. And I'm just going to take each of these little barrier boundaries, so these lines that have temperature markings, and just describe what I see. I haven't even looked at the question yet. I'm just writing what I think is important here. So we've got no cyanogenic or only acyanogenic. Below 4 degrees, or minus 4 degrees, sorry. Between 0 and minus 4 degrees, we can see reducing concentrations or reducing population numbers of the cyanogenic. And then as our temperature increases, so we start moving from 0 to 4 to 10 degrees Celsius, we see a massive increase in the number of cyanogenic plants, or the population that is cyanogenic. Above 10 degrees, okay, I can say that almost all of my plants are cyanogenic. Notice I'm not saying all of them, because this population here and this population here where I've put the red arrows still have some acyanogenic individuals. So just to kind of zoom out and just show you what I've written, I've figured out that cyanide is toxic to the cells of animals and plants. So that assumes then that toxicity means that they're going to be damaged, potentially dead, to the cells of animals, maybe these slugs, and plants, so the organism themselves that's releasing it. The membranes can be broken by slugs and cold temperatures. That's a bit of a problem. And cyanogenic plants with slugs or cold weather could cause the release of cyanide because of these enzymes being released from the cells that are broken. At temperatures below minus 4 degrees, I've got only acyanogenic plants, and there's also no slugs in this region. As I move from 0 to lower temperatures, the number of cyanogenic plants decreases. However, at temperatures above 0 degrees, I see increasing concentrations or populations of the cyanogenic plants. And at temperatures above 10 degrees, I can see that almost all of my plants are cyanogenic. So let's have a look at the question. Explain. So give reasons for the proportions of cyanogenic and acyanogenic plants in clover populations growing in the area where the mean minimum winter temperature is below 4 degrees and in the area where it is above 10 degrees. Cool. So this question now has given me specific temperatures to have a look at. And that is this section here, so where my orange little bracket thing is, and the section at the top. So, explain the proportions. I'm going to take each one of these in turn, and I'm going to describe what I see and then give reasons as to why the plants are proportionally found in each section. So firstly, I'm going to explain what I see. So below -4, all plants are acyanogenic, and above 10 degrees, oh, not 10%, almost all are cyanogenic. Cool. So why then, if we take these in turn, do we see the difference? The second mark is to suggest that below 4 degrees, or below -4, I'm going to keep making that mistake, the cyanogenic plants' membranes freeze. Okay, we know that when membranes freeze, they become much more solid due to that phospholipid bilayer, and this is going to have the effect of releasing cyanide into the tissues. If we're not sure where we got this detail from, remember in the stem we said that cyanogenic plants plus slugs or cold causes the release of cyanide, and we've questioned already whether that's going to affect the plant. Now this release of cyanide into tissues is going to be potentially damaging, and so actually what we see is a selective advantage, and we have to say that it's selective, it's a selection process. So it's a selective advantage to not produce cyanide at this temperature. And for your final mark, I'm going to put number 5 here, you could have said that the slugs aren't actually present here. So the slugs don't function at this -4. You could have also said that the slugs are present at higher temperatures, so the cyanide production is going to kill or deter them, and I guess that's going to be useful to link to this first mark. But broadly, you just needed to mention how having cyanide at -4 isn't actually going to help, it's actually going to hinder the growth. The diagram below shows pressure and blood flow during the cardiac cycle of a dog. Here on the top graph, I've got pressure in the left ventricle. I'm going to write down already that I know where I've got increased pressure. I'm going to have a decreased volume in that chamber, and this could be due to things like valves being closed or muscle contraction. Pressure in the aorta near the heart in kilopascals. Again, increased pressure results in a decreased volume. It could also be because I've got an increased blood flow here. So the blood is taking up all of the space and causing pressure against the side of the vessels. And we can see in this bottom graph the rate of blood flow in the aorta near to the heart. The increases in blood flow map to the peaks in pressure. Okay, so that's an important point to note. At P on the diagram above, so let me just highlight where P is. The pressure in the left ventricle is increasing. At this time, the rate of blood flow has not yet started to increase in the aorta. Use evidence from the diagram to explain why. And this question requires a little bit of knowledge of cardiac anatomy. Okay, so just as a little recap, the heart is split up into 4 chambers. We've got the right side on the left and the left side on the right. So we've got the right atrium, right ventricle, left atrium, left ventricle. And the aorta is a massive artery that loops round from the left ventricle over and actually compresses quite significantly onto this left atrium. Okay, use evidence from the diagram above to explain why. So I'm going to try and figure out what is happening in the aorta at the same time as those points P. So let's have a look, because the scale is all the same. So I can see that the pressure is the lowest in the aorta and that the blood flow is also quite low. Hold on. There we go. And so now I can see that the rate of blood flow in the heart, or in the aorta near to the heart, is low. So if I've got low blood flow in the aorta, this is probably because the semilunar valve that allows blood to leave the left ventricle is closed. Okay, in an exam, please be very mindful. I'm being lazy and I'm writing SL, but that's because I'm trying to do this video in an evening. You need to write semilunar in its full term. It does say do not accept SL. Okay, you have to write it fully. And this is closed because the pressure in the ventricle is less than the pressure in the aorta. Or if you want to flip that round, the pressure in the aorta is higher than the pressure in the ventricles. And just to double check, I can see on my graph that P is the pressure in the ventricle and is around, what are we at here, 8 kilopascals. Whereas in the aorta, we're seeing pressures at P of around 12 kilopascals. So the fact that the pressure in the aorta is higher than the pressure in the ventricles means that, sorry, this is a cross-section of the aorta. The valves are being forced closed. I'm going to put here the ventricles, because the pressure pushing back on that semilunar valve is greater than the pressure pushing from the ventricles upwards. At Q on the diagram, there is a small increase in pressure and rate of blood flow in the aorta. Explain how this happens and its importance. So this 2-mark question has 2 components to it, and you get 1 mark for answering each of them correctly. Okay, so explain how this happens. Well, we get a small increase in pressure and in rate of blood flow because the walls of the aorta are highly elastic. And the highly elastic walls of the aorta are able to undergo recoil. So this is a bit like when something stretches and then the force is let go, or the pressure is let go, and it kind of wibbles and bounces and it looks like it contracts a little bit, but it's actually just the elasticity sort of coming back to resting and then responding to pressure changes from that elasticity. What you cannot say for this mark, however, is that it's muscle contraction. It isn't. It's a passive process that occurs when the blood flow passes through the aorta at a specific point, and once all of that pressure of the blood has gone through the vessel, it kind of breathes a sigh of relief, lets go of all of that pressure that's pushing on the walls. The relaxing means that we get that recoil, that boing, if you like. That was a really good sound effect, if I do say so myself. Now, the importance of this, the second part of this question, is that the recoil means that we get smooth blood flow. If we didn't have this recoil, if we didn't have this increase in pressure and in the rate of blood flow, every time the heart beats, we would get a massive surge of blood and then no blood. Massive surge of blood and then little blood. Massive surge of blood when the heart contracts and then very little as we're in diastole. So this recoil and this slight increase in pressure, a little bit more of a humph for the blood, means that we get smooth blood flow, or the blood flow is maintained, and it also maintains the blood pressure. Remember, we've spoken about the blood pressure and the pressure the pressure is caused by the force of the substances, the structures in blood, so things like your white blood cells, red blood cells, platelets, proteins, all of that colliding with the walls of the vessel. Okay, and we want to keep that consistent. If our pressure drops or whatever, there's going to be some kind of physiological implications for it. And that's very important to maintain. A student correctly plotted the right ventricle pressure on the same grid as the left ventricle pressure in the diagram above. So again, quick recap. We've got the right ventricle on the left and the left ventricle on the right. The right ventricle is going out to the pulmonary artery and to the lungs, whereas our left ventricle is going, or is forcing blood out of the aorta to the body, so to the systemic circulation. Now what we need to do in this question is describe, so tell me what I expect to see, one way in which the student's curve would be similar and one way it would be different from the curve shown in the diagram above. So basically, what is the pressure going to look like in the right ventricle compared to the left ventricle? And this again requires knowledge of the anatomy of the double circulatory system that we see in the heart. So a similarity is that we are going to see, we are going to see peaks or the pattern of pressure being the same. And we know this because the impulse starts at the sinoatrial node, crosses the atria to form atrial systole, stops at the AV node, down the bundle of His, and causes contraction of the left and right ventricles at the same time. This forces blood out of the pulmonary artery and the aorta at once. Okay, that's really, really important. But the difference, and this is the second part of this question, the difference that we're going to see is actually that the right ventricle doesn't need as high a pressure. It's only going to the lungs. And at the capillaries of the alveoli, this mega high pressure could cause them to damage or break or just generally be a bit of a nuisance. We don't want bleeding in our lungs because of this massive pressure bursting everything there. So instead what we're going to see is that yes, the peaks and the patterns of the pressure changes are going to be the same, but the right ventricle is going to be at lower pressure. Okay. And the final question for this one. Use the information from the diagram to calculate the heart rate of this dog. And as if by magic, the graph has appeared. Okay, I'm only looking at this rate of blood flow in the aorta near to the heart. You could have used any of the other graphs. That would have been absolutely fine. But I've picked this one because it's got the time scale at the bottom. So the first thing I need to know is how long is one beat. Okay, and one beat is shown from the same, or the point of one wave to the same point on the next wave. And handily in this graph, they've mapped on 2 points for me, which are Qs. So I'm just going to draw 2 lines going down to my x-axis that show me where the Qs are. So I'm going to assume that this first Q, this line, is at about 0.23 seconds, and this second Q is at 0.59 seconds. Okay, so using maths, I'm going to identify that one beat takes the difference between 0.59 seconds and 0.23 seconds. And if I'm doing this in my head, which I am, that's 630. So that's 0.36 seconds, or roughly a third of a second. So each beat is a third of a second, and I need to figure out then how many beats I can get in 60 seconds. The heart rate is the number of beats per minute. So I'm going to do 60 beats per minute divided by the time taken for one single beat. And if I put that in a calculator, that gives me an answer of 166 beats, or 166.667 beats per minute. I'm not stupid, I'm not going to write all of that, so I'm going to change it to 166.7 beats per minute. That's much more reasonable and much more likely to fit in that graph. Obviously, there are a few different answers. The mark scheme has 3 different answers, and I'm going to assume those are for using each of the different graphs. So if you're using the rate of blood flow in the aorta to the heart, 166.7 or 167 beats per minute would have been fine. You could have also had, somehow, 164 beats per minute or 171 beats per minute. Again, not entirely sure. Haven't looked at all of the other graphs, can't be bothered. But there are multiple answers that you could have had for it. Ooh, question 7. I'm just going to give you the answers for this because we are going to do a revision session based on this process in a few weeks' time. So I'm not going to do too much about the nitty-gritty details. I'm just going to write down the answers for this one. So, describe the transport of carbohydrates in plants. First thing we need to realise is that the carbohydrate that we're talking about is sucrose. Please don't, in any transport of carbohydrates in plants questions, talk about glucose. It's never going to be glucose. Glucose is metabolised. When we've talked about photosynthesis, we've said that we're producing organic molecules, not just glucose. Okay, but for the purposes of translocation, it is sucrose only that we are talking about. And the first step of translocation is that we're getting that sucrose, and in our mark scheme it is underlined, it has to be involved, and it is actively transported. I'm just going to write AT, I am lazy. You need to write actively transported into, and we've got a few options here, into the phloem. You could have also said that it is actively transported into the sieve tube elements. And you could have also said that it is actively transported but co-transported as well. And again, I'm not going to go into too much detail about this. We'll do a revision session on it. But it's co-transported with hydrogen ions into the phloem by the companion cells. Okay, a companion is a friend, and these companion cells are friends to the phloem by helping load the phloem or the sieve tube elements up with loads and loads of sucrose. The effect of having sucrose in the phloem now is that we get reduced water potential in the phloem. Okay, we've got more dissolved solutes. That sucrose is a dissolved solute here. And what we see in the same mark point, reduced water potential in the phloem, results in the water from the xylem moving into the phloem by osmosis. We know that we have to mention osmosis. Any question relating to the movement of water, it's always best to just be safe and say it's by osmosis. And the impact of having this water now accumulating in our phloem is that we're going to get an increase of the hydrostatic pressure. So water being stationary in that place means that we're going to get that pressure building up locally. Okay, remember, it's only at that specific point where the sucrose has been loaded into the phloem. We get an increased hydrostatic pressure at that point, and that is going to produce a hydrostatic pressure gradient. So in places where there isn't sucrose, the sinks, for example, you're not going to get a high hydrostatic pressure because water is not going to move into those regions from the xylem, and so they're going to have a low hydrostatic pressure. We've got a difference now in different areas of the phloem of the plant. And what we see with this hydrostatic pressure gradient is that we see mass transport or mass flow of the sucrose to these sinks. And the sinks, you could have said, are to respiring cells. You could have also been nice and vague and said to storage organs, so things like potatoes. You could have also said to young leaves, fruits, seeds, shoot tips, root tips, or meristems. And the key detail about all of these is that these are active parts of the plant. Okay, so we're going to the cells that are respiring, they're using up the glucose or the sucrose or the carbohydrates. The storage organs are converting those monosaccharides and disaccharides into big polysaccharides, things like starch, to store them. Meristems are active, they're undergoing mitosis, they're producing more cells from the roots and shoots of the plant to build and make our plant bigger. All of these have low amounts of glucose in them or sucrose in them, because they're using them up. And so what we see finally at these sinks, these active regions of the plant, is that we get unloading or removal of sucrose into these tissues by active transport of assimilates. So the sucrose is called an assimilate by active transport, but you could have also said in the same mark point for this mark 6 by facilitated diffusion. Both of those are fab. So now we're getting into where the questions started getting better, I think. This question 6 was really, really well done. The reason why it wasn't well done is because some of you started to talk about the function, so things like a storage molecule or a structural molecule, where it's just asking to compare and contrast, so the similarities and differences of starch and cellulose. Do not tell me about what they do. So, the first thing I'm going to look at is the similarities. Okay, they are both polysaccharides. For the same mark, you could have said they're both glucose polymers or they're both made of glucose monomers. If they're both polysaccharides, for another mark, you could have said that they both have glycosidic bond. Bonds even, not glycosidic bond. Finally, you know that they're both carbohydrates, so they're going to have carbon, hydrogen, and oxygen. Now, you needed to make sure that you had at least one of these similarities to get 6 out of 6. Okay, if you've only spoken about the difference, you're not going to get full marks. It's a compare and contrast, so comparing and contrasting what is the same, what is different. You needed to have one of those 3. But then we get into the differences, and this was where you guys were really, really strong. It was nice to see. So I'm going to do a little table, and what you need to remember for these is it's a compare and contrast. So you need to say a feature of starch and then what the feature looks like in cellulose to give those full differences. Okay, remember it's structural. So the first thing I'm going to discuss is the monomers. Starch is made of alpha glucose, whereas cellulose is made of beta glucose. Lots of you spent a little bit of time talking about the inversion of the beta glucose monomers, so the 180 degrees rotation. It's not really relevant here. It doesn't overly matter. It adds nothing to the answer. But what does add to the answer is the shape of the molecules. So starch is helical, or you could have said it's coiled, whereas cellulose has straight chains. We're going to get into the fibrils and stuff later on. But another point that you could have said, so that is mark 4, that is mark 5. Mark point 6 is to say that starch is branched. But if you're going to say starch is branched, you have to say cellulose is not, or cellulose is unbranched. You can't just have that standalone sentence that starch is branched without mentioning cellulose. And again, looking at the straight versus helical chains, or helical shape, you could have said that cellulose forms fibrils, so those connections with hydrogen bonds, but starch doesn't. These are your main marks that were in the mark scheme, but you could have also, I'm just going to put a little dashy line to put the extra marks. You could have mentioned, and some of you did, about the bonding. So lots of you put that there were 1,4 bonds and 1,6 glycosidic bonds in the starch, but cellulose only has 1,4 bonds. And if you're not sure what this means, please go back and look at topic 1. As well, you could have said about the different structures that make up things like starch and cellulose. So let's put here structural things. You could have said that starch is made of 2 types of molecule. If you wanted to be specific, you could have said about amylose and amylopectin. You didn't have to. Whereas the cellulose is only 1 type of molecule. That was all that you could have had. Hopefully, though, there were lots of different mark points, but remember, you needed to have at least one of mark point 1, 2, or 3, those similarities between them, to get the full 6 marks. Alright, so final part of question 7 then. Another big one, 4 marks this time. Describe, so tell me how it's done, the complete digestion of starch by a mammal. And complete digestion means how am I breaking it down from its massive composite structure, this big polysaccharide starch, all the way down to glucose. And lots of you had fantastic ideas. Lots of you were so close to getting these marks. But remember, this is biology. We need the keywords. So I'm going to show you the marks. And obviously, you guys can see where you missed them. The first mark, just by itself, is to say the word hydrolysis. And you needed to say that the hydrolysis occurs with those glycosidic bonds. And where are these glycosidic bonds? Well, they're between all of the monomers that make starch up. So we're hydrolyzing the glycosidic bonds of starch to form a disaccharide. Okay, we're not going straight to glucose. This is really, really important. We don't get starch straight to glucose. It doesn't happen. But instead what we see is starch being hydrolyzed down to maltose. And this happens by the enzyme amylase. And again, loads of you were fab at saying this is in the saliva or that it's in the pancreas. And I'm not entirely sure why lots of you spent a lot of time talking about like the physical digestion, the mechanical digestion, using the teeth to increase the surface area and stuff. It's not overly relevant. It's not adding any detail to how starch is broken all the way down into little glucose molecules. Try not to waste your time. Stick to the question. So starch is broken down by amylase into maltose. We see hydrolysis of those glycosidic bonds, and then we see maltose is turned into glucose. If you want to be specific, you can say into alpha glucose by maltase. And the 5th mark that you could have said, so some of you did get 4 marks for this because you've mentioned all of these points so far, but you would have got a mark as well if you've said that these maltase molecules or the disaccharidases are membrane bound. Okay, so they're found on the membrane of the microvilli of the ileum, and this leads us really nicely into question 8. So cells lining the ileum of mammals absorb the monosaccharide glucose by co-transport with sodium ions. Explain how. Oh my goodness, this was absolutely terrible. So what I'm going to do is I'm going to draw it for you. Okay, so what I've got here is a visual representation of the ileum at the top. This is the small intestine. It's a hollow tube. Then I've got the epithelial cell, which I'm just going to write epithe. I can tell it's an epithelial cell. It's got those microvilli. It's got a nice big surface area to absorb substances. And then at the bottom, I have got a blood vessel. This is a capillary, but I'm just going to write blood. Cool. So, the first thing I need to realise is that in my ileum, I'm going to have lots of glucose. And I'm writing in there, I've got an increased concentration of glucose. And that is compared to the epithelial cell itself. So we know that co-transport is a passive process, but glucose is quite big, so we're going to need a second molecule, this sodium ion, and we need a concentration gradient. So how do I set up the concentration gradient for sodium to help me co-transport that glucose into my molecule? And the way I do it is by starting off forcing sodium molecules out of my epithelial cell into the blood. So mark 1 is that we're actively transporting sodium ions from the epithelial cell into the blood. This has the effect then of reducing the concentration of sodium inside my epithelial cell. Okay, so then mark 2, we get a concentration gradient of sodium ions between, now we're talking about between the ileum and the epithelial cell. So the sodium that was inside the epithelial cell has now been chucked out. There's sodium in the ileum. It's found in all sorts of food products. You know, salt is sodium chloride. So then we got that concentration gradient between the ileum or the ileal lumen and that epithelial cell. And so step 3 is now that this glucose is able to enter, wrong pen, the epithelial cell through co-transport. So the sodium is going to work its way down its concentration gradient. The glucose is just happy to be there and just comes through at the same time. And we need to finish this off for the third mark by saying glucose enters by facilitated diffusion. We can tell it's facilitated because we've got that carrier protein, that co-transport. We know it's diffusion because we've got that sodium concentration gradient that we've said in mark 2. And that facilitated diffusion happens with the sodium ions. So now we get into the questions that relate to this practical, where students set up the experiment shown in the diagram below. And we can see here that we've got some water in a beaker, we've got some visquing tubing, which the stem of the question has told us is partially permeable. Now already with this partially permeable visquing tubing, I'm thinking, okay, does it behave like a membrane? If it's partially permeable, does it mean that things can move through it? So movement of substances. And in my visquing tubing, I've got a mixture of starch suspension, so a solution of starch, and amylase. And we should know that amylase is responsible for breaking down starch into maltose, not into glucose. Okay, it turns it into those disaccharides. After 15 minutes, the student removes samples from the liquid in the beaker and from the liquid inside the visquing tubing. So she's taken 2 samples out and she carried out biochemical tests on these samples. She drew the table below to record her results. Complete the table by putting a tick in each box that you would expect to have a positive result. Now we're going to do each column separately. So we're going to do the beaker and then the visquing tubing. Before we get to there though, we're just going to recap what these tests are for. So the burette reagent is there for testing proteins. The iodine in potassium iodide is there to test for starch. And the Benedict solution is there for reducing sugars. So my beaker is going to have water in it, yes, and then we need to figure out whether there are any proteins present, whether any starch has gotten through, or whether any reducing sugars have been present. So we're not going to have proteins because they're too big. They simply won't diffuse. For the same reason, we won't have starch either. In the ileum, we might have other mechanisms, so things like facilitated diffusion or active transport that might move these bigger molecules off the membrane. But actually, they're not going to go through this Visking tubing. However, we are going to have some reducing sugars. That maltose is going to help, or that maltose is going to be able to be broken down and travel through the membrane. Inside the Visking tubing, we are going to have proteins, and I want to draw your attention to the fact that loads of you, if not all of you, got this wrong. Amylase is an enzyme, and enzymes are proteins. So therefore, you would have got this mark wrong if you hadn't said yes to this. For the iodine in potassium iodide, you would have got a mark if you'd put a tick in this or left it blank. Okay, so you could have said yes, there's still going to be some starch present, which means that the iodine would go from brown to blue-black, but equally you might have assumed that all of the starch has been broken down by amylase. So you could have had either of those. And then finally, yes, there is going to be some reducing sugars there because the amylase is doing its job. Helpfully, what we've just explained is the answers for question C. So justify your answers. Tell me why those answers are what you thought. So in the first case, if we focus on the burette reagent, you needed to say that the proteins are too large to pass through the tubing. So too large to pass through. For the same kind of answer as well, you could have said the iodine, except this time it's the starch molecules that is too large to pass through the tubing. But for the Benedict's test, you needed to say that the starch, and these are key terms now, the starch is hydrolyzed, not broken down, so it's hydrolyzed to maltose. And the maltose can then pass through the tubing. So it can pass through. In terms of marks, for this top table, you get 1 mark for each correct row. And then for this bottom mark, it does say that you get a mark for each of these standalone statements. But this Benedict's one says reject glucose. So you needed to be aware that the starch is broken down or hydrolyzed to maltose, not to glucose. Okay? The glucose comes after when we get to like membrane-bound disaccharidases and stuff like that. Thankfully and finally we are on to the last question, and there is evidence that the first photosynthetic organisms were primitive water-dwelling bacteria. The very first of these lived near the surface of the water in lakes and contained a purple pigment that absorbed light most strongly in the green region of the spectrum. Later, other bacteria evolved that lived on top of the sediment at the bottom of the lakes. Okay, so we've got our initial bacteria right at the surface of the water, and then some bacteria that have instead found a habitat at the bottom of the lake. Gene mutations, okay, let's think there already. I'm going to highlight this. This is important for me. Had enabled bacteria to synthesize chlorophyll instead of the purple pigment present in the bacteria living near to the surface. Chlorophyll absorbs light most strongly in the blue and red regions of the spectrum. So I'm just going to kind of annotate my diagram so I know what's going on. These first bacteria have got a pigment that is purple, and they can absorb the green light. Whereas the bacteria at the bottom of my lake have got the pigment chlorophyll, which we know is green, and they absorb the blue and red regions of light. So let's take a look at our graph and see what information we've got. And all this graph has done has told us what the stem told us, but in a data format. So I can see that my pigment-dwelling surface bacteria absorb most of the green light, and the chlorophyll sediment-dwelling bacteria have got 2 peaks in the blue and the red spectrum of the light wavelengths. The first question is a lovely, easy one. It is 5 marks though, so there is quite a bit of detail. Describe, so tell me how, light energy absorbed by chlorophyll molecules is used to synthesize ATP. So automatically I'm thinking, okay, I'm looking at the light-dependent reaction of photosynthesis. I know that the light-dependent reaction begins with the excitation of electrons. So excitation of electrons occurs. This is your first mark. The second mark is to say that the electrons are raised to a higher energy level. We know that once they're raised to that higher energy level, they become lost from the chlorophyll and move down the electron transport chain. So electrons are emitted from the chlorophyll. These are all individual marks as well, so all you had to do was write a nice, concise kind of series of statements, and the electrons are transported down the electron transport chain. While they're doing this and going down the electron transport chain, they lose energy. And that energy, which is where lots of you started at this kind of point, you spoke loads about the hydrogen ions and stuff like that, but all you needed to say was that energy, the energy that is lost by those electrons, is then used to synthesize ATP. And if you'd have thought about the question thoroughly, you probably will have mentioned about the electrons and stuff, and lots of you did pick up a couple of marks here and there, but lost like the full marks for this question. So part 7 is just to say that ATP is made from ADP and PI. Oh, pen's gone awry there. But hopefully you can see then, because we're specifically talking about how that energy, sorry, I don't know what's happened there, how that energy that I've absorbed by my chlorophyll has been transformed and transferred to the ATP. Yes, if you'd sat there and spoken about all of the hydrogen ions and stuff, that is true, but this is specifically asking about the energy. So it's used to excite, then that energy is lost, and that energy that is lost from the electrons is then transformed and transferred into those or that ATP synthesis. We've made it. We're at the end, guys. Last question. Using figure 2, explain how natural selection would favour the evolution of a sediment-dwelling bacteria containing a different photosynthetic pigment from those living near the surface of the water. And so what I've done here, I've just copied in figure 2. And I think what we need to realise is that with these, the bacteria that are at the surface are going to be taking up and absorbing all of those green wavelengths of light. So there's no more available for the bottom. But you can see on this spectrum, on the x-axis, that green is only kind of 1/3 of what we see. So in fact, the blue and the red wavelengths of light can reach the bottom of the pond or the lake or whatever to the sediment-dwelling bacteria. And this is where your marks come in. So the first thing you needed to say was that little green light, or a little bit of green light, reaches the bottom. And this is because it has been absorbed by the surface dwellers. Now, because of this, or in spite of this actually, the blue and the red pigments do penetrate, and they reach the bottom of the lake. Obviously, we know that natural selection begins with variation in a population. So variation of pigment colours in these sediment-dwelling bacteria has happened. Variation of pigment in sediment dwellers. As we're talking about natural selection, we see then that the bacteria that contain chlorophyll, and we're being specific to the question here, are able to absorb those red and blue wavelengths of light. They have a selective advantage. And then we get into our holy trinity of natural selection. They are able to survive, reproduce, and pass on the alleles. Remember, we're doing different versions of the same gene, so they've all got pigment genes. It's just whatever version you're expressing. Pass on the allele for chlorophyll. And over time, that is going to increase the frequency of this advantageous chlorophyll allele. So overall, we see this advantageous chlorophyll allele increasing in frequency, so its space in the gene pool will be more visible and more frequently occurring. Those that don't have the chlorophyll pigment are just going to die out because they can't absorb any of the other wavelengths of light. And with the end of that question, it is the end of the paper. The next question is the essay, but I will go through that with you in class altogether. I hope you found this useful. Bye.